This is the second part of metabolic pathways. So this video will cover inhibition and the different types of inhibition within those metabolic pathways. So as a quick introduction, what metabolic pathways are was discussed before, in the previous video. So they're enzyme controlled reactions, and metabolism is all the chemical reactions within your cell. So all of these pathways can be affected by inhibitors. So if you think about the general definition of the word inhibit, it means to kind of slow down or stop, okay, to get in the way of something. And that's exactly what inhibitors do. And because these inhibitors are getting in the way of metabolic pathways, they're essentially slowing down or stopping a pathway by slowing down or stopping an enzyme. Okay, so what you need to know is there are 3 types of inhibition. And I've got the first one on this side here on the left. Now that first pathway, and that first inhibitor, sorry, of a pathway, is called feedback inhibition. Now feedback inhibition involves an end product, okay, and particularly an end product when it gets to high concentration. So key detail, end product of this reaction would be at a high concentration. Okay, so in this reaction that I've got down here in this pathway, there's substance A being changed into substance B. Now that is carried out by, just for very general talking sake, it is enzyme number 1. Okay, substance B is then broken down into substance C, and that will be controlled by enzyme 2. And then substance C is changed into substance D, broken down, okay, by enzyme 3. So each of these pathways, parts of this pathway, are controlled by a different enzyme. So when feedback inhibition occurs, it's because the end product concentration is high, which means that this substance D is at high concentration. Okay, there's a lot of it. Now what that means is then that your cells are producing a lot of this substance D and it's not necessary. So what happens is this substance D will go to the enzyme early in the pathway, which in this example is enzyme 1, and it will bind to and it will block that pathway. It will bind to the enzyme and it will block the pathway. Okay, so in terms of your information aid, that inhibitor, that end product, which becomes the inhibitor, binds to the enzyme, and specifically it binds to the enzyme early in the pathway. So what that results in then is the pathway being blocked. Now that pathway isn't blocked forever, it's just blocked until this substance D starts to run out. And when it starts to run out, it's not able to then inhibit that enzyme 1. Okay, so it'll stop, so the inhibition will stop, that feedback inhibition will stop when end product concentration drops again. Okay, so that's the first type of inhibition. The key thing is that it will prevent further synthesis of this substance D. So when it binds to and blocks, it prevents that further synthesis of that substance D. So therefore it goes back and binds to this and it blocks this whole pathway. So the whole thing would then stop until substance D is then low enough in concentration that it's not there to block that in the first place. Okay, so the next one then is competitive inhibition. So just quickly draw a wee bit just to help me with time. So a competitive inhibitor, that is an inhibitor that has a similar shape. Okay, so similar shape or similar charge or similar affinity. Remember that meant attraction from before. to the enzyme as the substrate would. Okay, so what I mean by that is they're very similar. So they're very similar to the substrate. Okay, so they look very similar to the substrate, they are charged very similar to the substrate, and they have the very similar affinity as the substrate, and therefore they would compete with that substrate. Now if you think of an enzyme reaction, that substrate is trying to get to the active site, so it competes with the substrate, and specifically for the active site of an enzyme. Now because it competes with that, it binds to and it blocks the active site. The problem with that is then that the substrate cannot meet and cannot bind with the enzyme and cannot bind with the active site, and therefore it can't then go through the pathway that it normally would, and therefore that reaction is slowed right down. So I'm going to show you this in the diagram. So this would be our enzyme. Okay, and this is also an enzyme, but I'll come back to that in a second. And that enzyme might have a substrate. Okay, and we'll just put that in red. So this here is that enzyme's substrate. And that substrate is trying to bind to the active site. Okay, which is this area shaped here, and it'll look very similar in the previous video that I did. So that substrate will try and bind, but there's a competitive inhibitor that then gets in the way of that. Right? Now that competitive inhibitor then would come along and it would try to bind to the active site. Okay, so the competitive inhibitor will look similar in shape, similar in charge, similar in affinity, and all sorts of other things. So this one here would be your your competitive inhibitor. Okay, so that competitive inhibitor floats about just like the substrate would, and it will try to get to the active site of that enzyme before the substrate does. Now when it does that, what will happen is, I'll show you in this diagram here, that competitive inhibitor binds to that active site, and therefore because that competitive inhibitor has bound to that active site, the substrate cannot bind. Okay, so it's binding to and blocking that active site, and therefore the substrate can't bind to it and therefore can't be broken down or built up depending on the pathway. Now, what we can then do is we can overcome this inhibition. We can reverse this inhibition. And what we can do then is try and figure out how to do that. Okay, so this inhibition can be reversed. Now, if you think logically about this, there's one competitive inhibitor here and one substrate. So to increase the chances of your substrate getting to that active site and therefore binding, all you need to do is add more substrate molecules in, okay? And that gives them more of a chance of being the thing that will get to the enzyme first and therefore bind. Okay, so that prevents that competitive inhibitor then from being the first to bind. So inhibition can be reversed by increasing substrate concentration. So the more substrate molecules you add, and the higher the substrate concentration, the less likely it is that competitive inhibitor can then bind to and block that enzyme's active site. Right? So I'm going to show you the third type of inhibitor. So we've got feedback, we have competitive, And the third type then will be our non-competitive. So non-competitive works in a very different way from what competitive does. Non-competitive inhibitors do bind to the enzyme, but it binds to an area that isn't the active site. So it binds to an area of the enzyme away from the active site. Right? Now the result of that is then that if you're altering or binding to an area other than the active site, you then ultimately will change the shape of the active site. Now if we think back to National 5, if you change the shape of the active site, for example you denature it, if you expose it to high temperature or something like that, that denaturing changes the shape and therefore the substrate cannot bind. And it cannot bind to the active site. So if it can't bind, then therefore it can't be broken down or built up depending on the pathway, and therefore that pathway then stops. Okay, now it stops for that enzyme and it might slow down ultimately, and then it'll eventually only go as fast as it can possibly go for the enzymes that aren't affected. So in this diagram here, I've got another enzyme. Okay, looks very similar to the one I had before. And again, this area here is the active site. Now you'll know that because I'm going to draw the substrate on, and that substrate, the shape, would match that active site. Okay, it was trying to bind to that active site. So this is your substrate here. Now a non-competitive inhibitor won't try to match the shape of the substrate because it's not trying to compete for the active site because it's non-competitive. What it will do is it will try and bind to another area of the active, other than the active site, and therefore doesn't need to be the same shape as the substrate. Doesn't need to be the same affinity, same charge, okay? So this here is the non-competitive inhibitor. So if I show you the effect of that and draw the enzyme again, this time slightly different. Okay, so non-competitive inhibitor has bound to the enzyme, and because it is bound to the enzyme, it changes the shape of its active site. So now the active site shape is entirely different to what it was before. Now because of that then, the substrate, which has still remained the same shape because that's not affected, cannot bind. Okay, so this cannot bind to the active site of the enzyme. Now again, if you think about trying to reverse this, I could add in as much or as high a concentration of substrate as I like. In terms of me trying to get more reaction out of that, it's not going to happen. Okay, because my enzyme is destroyed, the active site shape is entirely changed, and therefore I cannot reverse that inhibition. That enzyme will not be able to be used. So this inhibition cannot be reversed by increasing substrate concentration. So what I'm going to do now is show you a comparison on a graph which compares competitive and non-competitive. Now in this graph here, okay, it shows enzyme activity on the y-axis and substrate concentration on the x-axis. So on an example question, you'd be asked to label which line, okay, was a competitive inhibitor and shows a pathway which has been inhibited by an inhibitor. Okay, which would be competitive and which would be non-competitive, so you'd have to pick between the two. Now you can tell this because of the effect of increasing concentration of substrate. So as this is increasing substrate concentration, on this purple line, it's having absolutely no effect. So the more substrate we add, it doesn't really matter to the enzyme. Okay, and that is because the enzyme has been changed. And the type of inhibition that would result in that is a non-competitive inhibitor. So this one here will be shown by a non-competitive inhibitor. Okay, and that's because as you increase substrate concentration, the enzyme activity doesn't get higher. It's always going to be low. It's not going to change because the enzyme now is not working any longer. And that means this one at the top is your competitive one. And that is because as you increase substrate concentration, you're making that enzyme more active and reducing the effect of that inhibitor. The more substrate you get, the less inhibitor can affect, and therefore the more active the enzyme can become. Okay, so this is affected by increasing substrate would reduce the effect of inhibition or the effect of inhibitors. And therefore that enzyme becomes more active. So enzyme activity increases. So that's summarised the three types of inhibition: feedback, competitive and non-competitive. In terms of diagrams, and if you're doing an essay question, you can absolutely use diagrams, but you must label them. Okay, in each point I've labelled the enzyme, like in this, and the substrate. And in previous ones I've labelled the active site too. I've labelled the non-competitive inhibitor. So make sure that everything in an essay question is labelled so that you don't lose obvious and straightforward marks.